Hi, my name is Suvi Sadaswam, and this is my research, Targeted Sequencing and Mutational Analysis of Follicular Variant of Papillary Thyroid Cancer. My principal investigator is Dr. Jason Bush, and my lead graduate student is Jasmine Cheatham. So, thyroid cancer is the most common endocrine malignancy in the U.S. with an average estimated 52,000 new cases yearly. And this is my research, Targeted Sequencing and Mutational Analysis of Follicular Variant of Papillary Thyroid Cancer. Of U.S. My principal investigator is Dr. Jason Bush, and my lead graduate student is Jasmine Cheatham. These are averages from about the past 3 years. So thyroid cancer is the most common endocrine malignancy in the U.S. Hi.
Sorry about that. Were you guys hearing double audio on that?
Yeah, we were.
Okay, I think. Okay, let me do that one more time. Sorry about that. Okay, let me give it one more try.
Hi, my name is Suvi Sadaswam Saxi, and this is my research, Targeted Sequencing and Mutational Analysis of Follicular Variant of Papillary Thyroid Cancer. My principal investigator is Dr. Jason Bush, and my lead graduate student is Jasmine Cheatham. So, thyroid cancer is the most common endocrine malignancy in the U.S., with an average estimated 52,000 new cases yearly. This is a brief overview of U.S. thyroid cancer statistics obtained from the American Cancer Society Statistics Center. These are averages from about the past 3 years. So we'll see that yearly death average is about 2,200, and this is maintained relatively stable over the past few years, even during the pandemic. Mortality seems to be at a steady level. However, over the past few years, the past year and a half, diagnoses have gone down because of lack of access to treatment facilities and cancer screening facilities. It's about an 80% decrease in access nationwide to these services. Still, our national yearly death average maintains over 2,000 deaths. So these cases are still prominent. These cases are, despite lack of testing and lack of access to care facilities. We can see that the number of new diagnoses for women outnumber the new number of diagnoses for men by almost 3 times. And additionally, California continues to have the highest incidence of thyroid cancer in the US. So these are just a model for the different types of cancer cells, and what we're looking at for my research is a differentiated type of cancer called follicular variant of papillary thyroid cancer. So here's a flowchart depicting the different types of cancer, the different types of thyroid malignancies. Essentially, differential cancer cells are cancer cells that look like harmless cell tissue, and undifferentiated cancer cells look very drastically different from their surrounding cell tissue. So they look and behave very differently, and it makes it very easy to tell under a biopsy which cells are harmful and deadly. So, let's see. Brings us to our next slide. Now, under biopsies, these are what the different types of cancer tissue look like. So on our left here, we have follicular variant of papillary thyroid cancer, which is what I'm looking at, FVPTC. And then our middle slide is PTC, papillary thyroid cancer. And our third slide is follicular thyroid cancer. So in the early 90s, it was believed that FVPTC was a small component of papillary thyroid cancer biopsies. Now, over 50% of differentiated thyroid cancer biopsies consist of FVPTC, making this the second most common differentiated thyroid cancer type. Now, the major issue now we'll be discussing in the next slide is which lesions of FVPTC are benign and which have the potential to metastasize. So the distinguishing criteria for pathologists to diagnose and determine which lesions are actually FVPTC is still highly debated upon. So it's difficult since this is such a subjective way of diagnosing FVPTC. It differs and varies from each pathologist to the next. So what my research attempts to do is find an objective way, a way to look at DNA and RNA and have us help decide which of these lesions are actually harmful to help pathologists and to help diagnoses later on. So our initial approach was 1, to obtain tumor tissue, 2, to stain our tissue slides, 3, laser capture microdissection, 4, DNA and RNA extraction and quantification, and 5, evaluation of RNA. So basically reading our RNA and our data and determining what we have. So here's a brief overlook at different tissue slide staining. This is what our slides look like initially at the top, and below is what our tissue slides look like after staining. Now these areas of the tissue have been marked by one pathologist so that we could have consistent samples of which parts of this tissue, this thyroid cancer tissue, is actually harmful and malignant or cancerous, and which is benign, so the one that's not going to metastasize. This is our protocol, and staining these cells allows us just to basically look at it under a microscope more clearly. So laser capture microdissection, we have a very convenient laser capture microdissection machine in the biology department that allows us to not only look at these slides in very close detail, but it also allows us to make very specific cuts into our tissue slides. So on the left here is our slide before our cut, and on the right is our slide after we have made our 2000 micrometer cuts. And that allows us to basically get tissue that is control, which is the non-malignant tissue, the non-cancerous tissue, and then separate that from our tissue that is actually cancerous so we can differentiate between the two genetic samples. So this is our one-step PCR machine, is our thermal cycler, our RT-PCR thermal cycler, and that allows us to amplify genes of interest via PCR. So this is what we look at after we've amplified our PCR. So normally we would like to have very strong dark light bands in this case. However, our data is very fuzzy and dull. And that's because RNA degrades very quickly, making it really difficult to obtain significant data. So how are we going to address this? So this is what we're going to do in our next steps. So we had to take a step back for this, and a lot of times in scientific research that this is what it requires, is to look back and see what possible areas we can improve on. And so we realized that DNA is way more stable than RNA, and that maybe this is the better route to go down. So what we decided to do was do targeted sequencing of cancer DNA, specifically the NRAS oncogene DNA, which is the cancer DNA associated with FVPTC. This has been heavily associated with thyroid cancer malignancies in recent years, and we decided that we're going to analyze this DNA, this long strand of code, break it down and determine the different nucleotides and how many base pairs are in between each exon. So afterwards, we can amplify these different genes and determine if there are any mutations among the different samples that we currently have in the lab. So here are my acknowledgements. Thank you to Dr. Jason Bush and Jasmine for leading this research. Please let me know if you have any questions. Thank you.
Okay, we can go ahead and get started with the Q&A.
Great. Dr. Brooks. Hi, good morning.
Great. It was a great talk. It's very interesting. Thank you. I just wanted to kind of ask a little bit about this NRAS gene that you're going to be looking at. So, you know, this like cancer diagnosis thing is always very interesting to me because, you know, if you don't have 100% of the things having the thing you're looking for, then you always have the risk of, you know, missing the diagnosis. So I guess I'm just wondering how, you know, frequent is it that this NRAS gene is mutated in these types of cancer, or whether that's even known?
Well, in the past few years, we've noticed a trend in thyroid malignancy testing, and previous scientists who have done published works on follicular variant thyroid cancer have noticed a high association between NRAS oncogene at exons 2 and 3 and their association with various thyroid malignancies. Now, what we don't know is how frequently those mutations in NRAS are associated with my type of thyroid cancer, which is FVPTC. We know that they're very highly associated with thyroid malignancies these past few years, but we just don't know how frequently and how often. And specifically with our samples of DNA, our samples of thyroid cancer, which we've acquired from Central Valley patients. It'll be very interesting to look at and be able to draw a conclusion and see how frequently this mutation is associated with this type of thyroid cancer. And it'll help pathologists later on determine and see if people are predisposed to this in their genetic code somewhere later on, besides waiting until it gets later on and it becomes malignant. And then we can test for that very early on and be able to go on a diagnostic route that fits the patient.
Sure. Do you mind if I just ask one follow-up question then?
Yes, of course.
You know, that's interesting. So like really you're trying to see whether there is like a causal or a link, right? To see, you know, if I look at 1,000 cancers, you know, how many have mutations in that gene or whatever. Do you have a sense of how many, you know, statistically, how many samples you'd actually need to look at in the long term to know for sure?
Oh, there's very great implications for this type of research. And currently we only have access to 20 patients in the Central Valley. So being able to see that link would be great. But later on, I would love to see at least 1,000, possibly way larger sample size than that, to be able to look at the correlation between this. We haven't looked at statistics and how many samples would make it significant, but so far we do only have 20 thyroid cancer slides to work with.
Okay, thank you.
Thank you so much.
Thank you for that. So we're going to go ahead and get started with our next presentation. Just a quick little reminder, I'm going to be starting the videos about every 15 minutes is when the next presentation is going to start. And again, if I cut you off, no worries, that's okay. I'll try and see if there's any time at the end for any other Q&A if we have a little bit of time left. So let me go ahead and get started with the next one. And the next presenter is Esveidi Oseguera.
Hi everyone, my name is Esveidi Oseguera, and today I'm going to be presenting natural products for the potential treatment of triple-negative breast cancer. It's also known as TNBC. So what is triple-negative breast cancer? There is common estrogen and progesterone receptors known to scientists that are not found in TNBC, and they don't make too much protein called HER2. HER2 is a growth promoting protein on the outside of all breast cells. So breast cancer cells with higher than normal levels of HER2 are called HER2 positive. And these cancers tend to grow and spread faster than other breast cancers, but they are more likely to respond to treatment with drugs that target the HER2 protein. Unfortunately, in TNBC, HER2 is negative, and hormone therapy and drugs that target the HER2 protein are not helpful in treating these cancers. Triple negative breast cancer accounts for 10 to 15% of all breast cancer. And due to the HER2 being negative, it limits the treatment options and has a worse prognosis. So the only treatment available at the moment is chemotherapy, which is really hard on the body and natural normal cells that are healthy for us. So TNBC is the most aggressive type of breast cancer due to the rapid growth that it has. It is more common in women younger than 40, and it is more likely to have spread when it is found, and it is more likely to come back after treatment. So what are natural products and why are they useful? So natural products are chemical substances found in nature, and they have distinctive pharmacological effects. They can be extracted by living organisms, or they can even be prepared by total synthesis. Natural products have long been proven to be good lead compounds for the treatment of various cancers. So in this study, I have been researching numerous natural products that have reported to have potential in treating TNBC based on the data reported in the cell and animal models. And overall, I found that Lysyl-Chalcoin A, LA, was the best, and it was chosen as our lead compound in this project due to the relatively greater potency and synthetic accessibility. So to optimize LA, the structure is shown on the right, we wanted to engineer its analogs to enhance the potency in inhibiting triple negative breast cancer cells. And to do so, we created a group of analogs that LA can have been designed by replacing the hydrophenyl located on the left, the circled in the red, in LA with the 1-alkyl 1H imizadotol in hope to improve its potency. And here it is shown on the right, also circled in the target analogs. So the synthetic scheme. So in the first scheme, we wanted to produce the 9 target analogs, 1 through 9, to be synthesized by the aldol condensation of aldehyde 10 and ketone 11. So here, aldehyde 10 and ketone 11 will be added together to produce these target analogs, and they will vary in the R group below. So in scheme 2, we wanted to, compound 12, shown on the left, will undergo Claisen rearrangement, and this will produce aldehyde 10. Then we wanted to produce the synthesis of ketone 11, and we will start by having aldehyde 14, and it will be protected as a tosyl group in compound 15, as seen here. And then this will react with the Grignard reagent, MEMGBr, to give ketone 16. The deprotection of the ketone 16 with piperidine will give ketone 17. So ketone 17 will react with the appropriate alkyl halide, RBr, meditated by potassium carbonated to produce ketone 11. So how will we know if this is successful? So the chemical structures for all the intermediates in the final products will be characterized by hydrogen NMR, carbon NMR, IR, HRMS, and their anti-proliferation activity against the two triple negative breast cancer cells lines, which is MDA-MB231 and MDA-MB468, will be evaluated using WST1 cell proliferation assay. So what's the difference between these two cancer cell lines? So MDA-MB468 is a basal breast cancer cell line, while MDA-MB231 is a Claudin-low cell line. So in MDA-MB468, they're most closely maps basal tumors. And while MDA-MB231 closely maps not basality, but rather the new Claudin-low. So they are vastly different in molecular and phenotypic behavior, with MDA-MB468 being a highly proliferate and prognostically highly compromised relative to MD-MB231, which does not have highly proliferate. So what happens next? In the months of April to May, we will begin the synthesis of shown before. In the months from May to June, we are hoping to analyze and summarize the data that we have found and hopefully publish it. So I would like to express my special thanks of gratitude to my PI mentor, Dr. Chen. Also, this material is supported by the NSF under this grant and the Chancellor's Office of the CSU and Fresno State. The references I used to produce the synthetic scheme are shown here. And does anyone have any questions? Thank you.
Thank you so much for that, Isbaby. I'm going to open it up for questions now.
Hi, good morning, Dr. Hawker.
Hi there. Thanks so much for your talk. Can you talk a bit about your thinking about the next steps and also the previous research in terms of measuring the potency of these molecules? Can you talk a bit about that?
Yeah, so previously, due to COVID, I wasn't able to get into the lab and actually do hands-on research. So I was doing literature research online, and I was looking for different natural products and comparing the potency of each. And so we found that lycopene A was the best one because of the values that we found in each article, and also like the synthetic accessibility of that compound.
Got it. And when you say potency, how is that determined?
So we look at the ICR values, I believe. The values after they treat triple negative breast cancer cells with these natural compounds in these articles. And we determined just by comparing like how easy it was to complete like this, to complete this experiment, and how like we can be able to recreate it and make it better.
Hi, good morning. May I ask one question?
Yes, go ahead.
So could you explain the more like a detailed reason why you choose this specific analog? What is the reason behind that?
So we're hoping to find like a good target analog. These are just like different types of analogs that we hope will be successful. So we don't know if any of these will be successful or not. But like hopefully, because there is 9, we will find one that has a higher chance of producing better results than previous studies.
Thank you.
Okay, if we have no other questions, I'm going to go ahead and start connecting our next presenter, who is Jorge Jacinto. Let me get that one started.
Hi, everyone. My name is Jorge Jacinto, and today I will be talking about my research, which has been on the molecule beta-N-methylamino-L-alanine, and specifically on how I've been using computational chemistry techniques in order to compare its geometry to that of glutamate. So before I get started on showing some of the data that I've been able to gather so far, I wanted to give a brief introduction on this molecule and the motives that we have for studying it. So beta-N-methylamino-L-alanine, also known as BMAA for short, is a non-protein amino acid which has long been associated with the onset and progression of the disease complex known as amyotrophic lateral sclerosis Parkinsonism dementia complex. BMAA is produced by various types of cyanobacteria, and it is believed that high exposure to BMAA from the consumption of cycad seeds was one of the main causes that led to the elevated incidences of this disease in the native people of Guam. On top of this, BMAA has also been implicated in the onset and progression of other neurodegenerative diseases, since previous studies have shown its presence in the brain tissues of patients with Alzheimer's disease and ALS. The problem here is that despite these strong connections between BMAA and neurodegenerative disease, its exact role is still not clearly understood. So there have been many in vitro studies that have shown that BMAA is indeed detrimental to neurons, and recent studies demonstrated that the presence of bicarbonate ions is essential for BMAA to gain this neurotoxic functionality, which is important because bicarbonate ions are an essential component of physiological systems and help maintain the pH. Further studies found that this presence of bicarbonate ions leads to the formation of primary and secondary carbamate adducts of BMAA, where CO2 molecules produced by bicarbonate react with either the alpha or beta amine groups of BMAA, leading to the formation of these two carbamate products shown here. In particular, we are interested in the beta carbamate products shown here on the right, because if you look at it closely, its structure very much resembles that of glutamate, especially since both of these molecules contain both an alpha and beta carboxylic acid group. So these findings have led researchers to hypothesize that the mechanism of BMAA neurotoxicity involves cell death caused by the overactivation of excitatory amino acid glutamate receptors. So excitatory amino acids normally activate these types of receptors in order to regulate synaptic transmission, but it is also known that overstimulation leads to neuronal apoptosis. So for my project, I wanted to take a closer look at these structural similarities. Unfortunately, due to the situation with COVID, I was not able to start working in the research lab and do real experiments as I planned. So for the meantime, I had to resort to using computational methods to collect data. So the research question that I wanted to address for this provisional computational study is, how likely is it that this similarity in structure between the beta carbamylated BMAA and glutamates will allow BMAA to bind to and overactivate glutamate receptors? And as I mentioned, this study relied mainly on computational methods to collect data for the meantime. And the main programs that I've been using so far to investigate this research question are IQML and QChem. IQML is an open source visualization and molecular editor package, which can be used to interface with QChem. QChem is a general purpose electronic structure package featuring a variety of methods which are implemented using innovative algorithms, which enables fast calculations of large systems on various computer architectures. These calculations are performed using density functional and wave function based approaches. And this program offers a lot of useful tools, including a large selection of functionals and correlation methods, methods for electronically excited states, solvation models, and wave function analysis tools. In this study, IQML was used to first construct the molecules of interest, and then to set up specific calculations for QChem to compute. So here I just included an image of kind of what the QML interface looks like. So here you can draw the molecules, and then you can set up calculations, and there's a bunch of different tools that you can use that are very useful. So the main calculations that I've been able to perform so far are geometry optimization calculations, which to me were some of the most important ones because I wanted to see whether or not the similarity in structure was going to cause BMAA to also adopt a similar geometry to glutamate, which would indicate that it's possibly more likely to bind to the glutamate receptors. And then I also calculated the vibrational frequencies in order to see if the geometries that were optimized represented real stable structures or transition states. So the research hypothesis for the study is if BMAA truly causes neurodegeneration via overactivation of glutamate receptors using its structural similarities, then the beta carbamylated form of BMAA should adopt a very similar molecular geometry to glutamate. So the initial geometry optimizations for beta carbamylated BMAA and glutamate showed that both of these molecules adopt a very similar geometry. So shown here are visual depictions of both of these geometries as shown on IQML. On the left, we have glutamate, and on the right, we have the beta carbamylated BMAA. However, for some reason, I was having problems when trying to run more calculations on these two molecules specifically, since my computer would usually crash when sending the calculations to the QChem server. It seemed that the program was having difficulty with converging when setting up the molecules in their charged Zwitter ion forms. And the reason I set them up this way is because these are the forms that would predominate at physiological pH. So I decided to run calculations using the uncharged forms of these two molecules instead, and I had an easier time getting the optimizations to converge. So here's visual depictions of those molecules, namely glutamic acid and BMAA, with the carboxylic acid groups protonated and the amine groups deprotonated. So overall it has zero charge, and it seemed that the program was having an easier time running calculations on these molecules. And shown here is a side-by-side comparison of bond lengths and bond angles for glutamic acid and beta BMAA. The values shown in this bar graph are averages from many geometry optimization calculations that I ran, and they demonstrate that there is very little geometric difference between the two molecules as was calculated by QChem. So despite having problems when running calculations for the Zwitter ion forms, I decided to also compare the average bond lengths and bond angles using the few calculations that I was able to run for the charged molecules, glutamate and BMAA, with the amine group protonated and the carboxylic acid groups deprotonated. And similarly, we see that there's very little difference in the bond lengths and bond angles between glutamate and beta BMAA. Shown here is once again are the differences in bond lengths and bond angles for these two sets of molecules, this time with the differences plotted in a line graph instead. I thought this would be useful in particular because it's easy to notice that the bond angles and bond lengths are much more similar towards the ends of the molecules. This is reassuring because it is the alpha amino region of glutamate that is known to interact with glutamate receptors. Seeing that the geometries are extremely similar towards the end region, as we can see here and here on both of these plots, overall it supports the hypothesis that beta BMAA should adopt a similar geometry to glutamate if it is to overactivate glutamate receptors. One thing that I should mention is that all of these values were obtained prior to running the vibrational frequency calculations. Once I started going over the data for the vibrational frequencies, I realized that many of these structures actually represent transition states, since many of them had negative or imaginary vibrational frequencies. Nevertheless, the results are still supportive of the theory that BMAA may cause neurodegeneration via the overactivation of glutamate receptors, especially since the chemical environments and physiological systems are likely to influence the geometries anyway and change the conformation of these two molecules. So for future directions, one thing I'm currently doing is I'm using IQmol to optimize the geometries of Zwitter ions in the presence of a solvent. So that's another thing that IQmol can do. You can set up the dielectric of the solvent and run calculations that way. I'm hoping that maybe I can get them to converge more easily in this manner. And as I mentioned, I'm also analyzing the data for the vibrational frequencies currently. Another thing I want to do with IQmol is compute the energies in the presence of different metal ions. And this is because some studies have actually shown that the presence of certain ions can shift the equilibrium of this reaction that I showed earlier in either direction, and that they can bind to certain of the carbamates of BMAA with higher affinity. So it'd be interesting to see which of these is more stable as calculated by QChem. Another thing that I want to do is calculate the NMR shifts using IQmol. And I'm also trying to learn how to use this other software called Autodock Xena in order to simulate docking of BMAA to glutamate receptors. So hopefully I'm able to do that. And then hopefully in the summer, once I can actually start working in the lab, I can conduct some real in-lab NMR experiments with BMAA and glutamate in different solutions and in the presence of different ions and go from there. So thank you for listening, and I'd be happy to take any questions you may have.
Okay, we can go ahead and get started with the Q&A. Go ahead, Dr. Brooks.
Hello.
Hi, great. That was an interesting talk. So I'm kind of curious about the glutamate receptors themselves, right? There's a bunch of different ones, if I recall. Is it known whether the BMMA, is thought to interact with all of them, or some subset of them? Is there any kind of information about that?
Not that I know of. So that's kind of what I'm trying to look at here, see if based on the geometry, if I think that's going to be able to. From what I read, I didn't find that there's any experiments showing that it can bind to them. The ones that I was looking at were the NMDA receptors, which I believe from what I read were the ones that are known for being overactivated and causing excitotoxicity, probably among other ones as well. But I mean, this information presented here is just very preliminary. I'm just trying to show that it might be able to adopt a similar geometry and then kind of go from there. And maybe use the information I learned here to actually set up some real experiments once I'm able to work in the lab.
Sure. I guess sort of like a follow-up is, I definitely don't know a ton about these, but I seem to remember also there's a lot of different like agonists and antagonists, right, that bind to these things. So they interact with more than just glutamate in the natural setting. Have you thought about comparing, the structures of the agonists and antagonists to BMMA as well to see if there's something you can learn?
No, I have not done that. That is a good idea. I was looking at other amino acids because I know there are some other molecules and amino acids that activate these types of receptors as well. So that was going to be my next step, to maybe try to do these same type of calculations for other molecules that may be analogs or maybe antagonists of this type of receptor as well. So yeah, that's a good next step.
Great, thank you.
Thank you. Do you have any other questions? Yes.
Go ahead, Dr. Acker.
Thanks so much for the talk. I wanted to hear a little bit more about how you think that the findings for those uncharged species that you found relate to kind of your overall research question, and how do they kind of work in conjunction with your other findings for the more, I guess, physiologically relevant charged species?
Right. That's a great question. So, I mean, the issue here is I've been having a lot of trouble running these programs. I have a Mac, and it's been taking me longer to actually run these calculations, and then the program sometimes crashes. So it seemed that when I was running the calculations with the charged molecules, the program would crash. That maybe the calculations were too long. I'm not sure what was going on. One thing that I noticed is that I seem to keep getting transition states, and the amine would try to transfer a proton to one of the carboxylic acid groups. So that's what it seemed like. The molecule was kind of shifting, and that was going on, and then it wouldn't converge. It would just stay that way. So that's why I decided to run the calculations on the uncharged versions. And those, I had a much easier time getting the calculations and the geometries. And then, so I got those, and I was able to get a lot of averages for those. And then once I saw that that was working, I tried again with the uncharged, and I got some calculations to finish through. So I guess to answer your question, the reason I think it's relevant, I'm just comparing it. And if you saw those graphs that I showed, they kind of showed the same trend, the charged molecules versus the uncharged ones, in terms of the differences getting bigger towards the middle. So, I mean, my goal would be to really run all the calculations on the charged molecules, but I just kind of wanted to get started and start to try to show some data with that. Yeah, that's a very good question.
Thank you. Thank you.
Thank you.
Thank you for that. Jorge, we're going to go ahead and I'm going to start setting up our next presenter. The name is Keaton Montgomery.
Thank you.
Alrighty folks, my name is Keaton Montgomery, and the title of this project is Spatiotemporal Metabolomics of Amazolo Treatment in Fukumoto Navel and Clementine Mandarin Oranges. Proton NMR metabolomics. This is the experimental overview of your typical proton NMR metabolomics study. What is metabolomics? Metabolomics is the study of the metabolite composition of your targeted biological tissue. For plant metabolomics, the targeted tissue can range from the actual leafy tissue, the woody tissue, the roots, the actual agricultural product, or even the different layers of your agricultural product.
It's 9 o'clock.
The experiment can be either observational, in which you're just trying to acquire your metabolite composition of your samples, or it could be manipulative, in which you add a treatment effect and compare it to a control. So tissues would need to be isolated and processed for data acquisition. A solvent that is compatible for metabolite extraction would need to be used. The solvent would need to be removed via lyophilization, leaving only our freeze-dried samples with our metabolites. And the freeze-dried samples would need to be suspended in a solution containing a compatible solvent for NMR spectroscopy and an internal standard for quantification. Once all of your samples are ready, data acquisition needs to be done utilizing NMR spectroscopy. Here at Fresno State, we utilize NMR spectroscopy for our data acquisition for our metabolomic studies. Once you gather all your data, you want to do your statistical analysis and pathway analysis. Now, to introduce the citrus industry, this plot represents the amount of fruit produced annually for several agricultural products. So for citrus, a total of 100 trillion pounds are produced annually. This is quite significant. It requires a huge workforce of harvesters to harvest this fruit every year, a huge workforce of truck drivers to transport fruit to the pack house for processing and to the market, and even a huge workforce of pack house workers to process this fruit to suitable conditions for the market. So basically, the citrus industry is a huge economical contributor not only to agriculture but as a whole. As with any other agricultural practice, you're going to face some economic losses. Here are some to name a few. Post-harvest pathology. Typically, a pack house will apply fungicide after harvest to cure the infections that were induced during harvest. If this isn't done, a 50% reduction in yield is estimated, which is quite significant. Imazalil is commonly used as the fungicide. It works well and it's cheap. Relatively cheap anyways. And Penicillium digitatum accounts for roughly 50% of these post-harvest pathogens. It's one of the main culprits. As for market conditions, let's say for instance there's a certain year where citrus isn't selling. That means fruit will either just stay on the tree or stay in a cold storage room in a pack house. And the fruit is just getting older and older and older, and you're going to lose a lot of product due to overmature fruit. Now, fruit maturity is assessed in the industry, and it is assessed as total soluble solids and percent acidity. So the goal of this research, we know Imazalil works well. We know it does a good job at what it needs to do. But we don't know how it actually alters the physiological tissues of our citrus fruit when treated. And as for fruit maturity, we know that total soluble solids are assessed, but we want to identify specific metabolites associated with fruit maturity. So the goal of the research is to develop a comprehensive analysis of citrus fruit, as well as identify biomarkers and metabolic pathways associated with fruit that is treated with Imazalil, biomarkers associated with fruit maturity, and biomarkers associated with the different tissues of different commodities of citrus fruit. Here is our experimental design. 2 different commodities were assessed, navel and clementine, and the 3 different tissues were assessed. The flabido, the outer leathery layer of the rind, the albedo, the inner spongy layer of the rind, and the juice. Fruit was harvested here at Fresno State. And we have our control, 5 reps of our control, in which the tissues were processed immediately after harvest. The tissues were isolated, metabolites were extracted, and the samples were processed for data acquisition. We have our first treatment, 5 reps of our cold storage treatment, in which fruit was harvested and then placed into cold storage for 10 days at 40 degrees Fahrenheit. After 10 days, the tissues were isolated, metabolites were extracted, and samples were processed for data acquisition. And then we have our third treatment, our cold storage treatment with fungicide, in which the fruit was harvested and then treated with Imazalil in a little dip bucket in the lab. The concentration utilized is the concentration that is widely utilized within the industry. After fungicide treatment, the samples were placed into cold storage for 10 days at 40 degrees Fahrenheit. Tissues were isolated, metabolites were extracted, and these samples were processed for data acquisition. So after all these samples were processed for data acquisition, NMR spectroscopy was utilized to gather all of our data, and Keynomics was a program that was utilized to identify all metabolites in our samples and to quantify those metabolites as well. As for our results, a total of 27 metabolites were identified, 4 sugars, 10 amino acids, 7 organic acids, and 6 other metabolites. Other just meaning it doesn't belong to one of these 3 categories. 25 metabolites were common within the literature, and 2 of the metabolites identified were not identified in the literature. So to assess metabolites associated with fruit maturity, null hypothesis significance testing was conducted comparing cold storage fruit to our control. So these are 2 tables, one for each commodity, navel and clementine, and the 3 different tissues of each, associated with the fold change and p-values. P-values of less than 0.05 are highlighted in red, representing significance, comparing a significant fold change comparing the cold storage fruit and the control. As for the fold change, look at arginine, for instance. There is a minus 1.63 fold change in arginine concentration in the albedo tissue with respect to comparing the cold storage fruit to our control. So basically, many metabolites were identified associated with fruit maturity, which can serve as potential biomarkers to assess fruit maturity. Another noteworthy take-home point is that these two different commodities are assessed in the exact same way within the industry, but they each had a different response as the fruit matures. Comparing cold storage versus our Maslow treatment to identify metabolites that are affected via a Maslow treatment. And the same type of tables, and again, we have identified many metabolites that were altered due to a Maslow treatment. So in conclusion, proton NMR spectroscopy can quantitatively differentiate between the two commodities. It can quantitatively differentiate between the three tissues, and can detect metabolic responses associated with a Maslow treatment and fruit maturity. A total of 27 metabolites were identified. 20 plus of these can serve as potential biomarkers associated with fruit treated with a Maslow or fruit maturity.
Thank you. Thank you so much for that, Keaton. We're going to go ahead and open it up for any questions.
Yes, sir. Dr. Yoshida.
So first of all, could you clarify, is albedo is the spongy layer or flavido is the spongy layer?
The albedo is the white portion of the rind that's kind of spongy.
Mm-hmm. And then flavido is the most outer layer?
Yes, sir. The leathery, tough portion of the rind.
So just before I hold your presentation, I expected you to see the most difference in the flavido outer layer, regardless of the type of the oranges. But when I look your comparison between cold storage versus the treated with these chemicals, I see one probably clementine. There's some difference between these two conditions on albedo, and then navel orange, you see the difference in flavido layer. So is there any reason why, for example, the navel orange, you see the most significant difference in flavido versus the clementine, you see the significant difference in albedo layer?
You know, that's a very interesting finding because the navel rind is much thicker. So I was actually expecting to not find as much interaction in the navel rind compared to the clementine rind, which is very thin. But we actually found much more activity in our the navel rind, which was just completely unexpected. As to explain why, I honestly have no idea.
Okay. Thank you.
Sir. Yes, sir. Dr. Brooks.
Great, Keaton. Thank you. That was pretty interesting. So I guess I'm just kind of curious. You know, I think we talked about this in your defense as well. So I don't want to, rehash things we already talked about. But I'm just, obviously like using NMR in the field, right, is not like practical. And I think you talked about it before. I'm just kind of curious whether you think, so I guess like I'm wondering about the genetic information. So, is the genomes and things of these oranges known, or has that been sequenced? And like, so if it has been, you could potentially take the information you have here, right, these metabolites, these that are being produced, and then presumably, genes involved in producing those metabolites or, you know, or not producing them, right, depending on what you're looking at, or either being turned on or off. And that'd be a much easier thing to look at in the field.
You're right. The genome has been produced and has been and it is available in pathway and certain libraries. And that is one thing that needs to be developed to actually identify certain conditions that are practical. That would probably be a better route in the actual industry to identify specific genes that are upregulated or downregulated, opposed to actually developing methods to identify a specific metabolite associated with a condition of interest. So that would probably be the better route choice.
Thanks. Actually, just kind of like a curiosity question related to that is, are most economically important crops are the genomes known, or is it just kind of a handful of them? Or do you
I'm not 100% sure on that one.
All right, thank you.
Yes, sir. Dr. Hucker.
Got about 1 more minute, guys.
Okay, I'll be quick. Hey, Keaton, thanks for the talk. I just wanted to ask about the methods that you used in terms of you presented both the overview and also in terms of the tissue, collection and dissolution, I guess, for lack of a better word. Can you talk a bit about that method in terms of its standardization across this type of research, or is this just like a method that you developed independently?
We kind of took bits and pieces through different literature throughout the literature, and we tweaked it. We had a pilot trial about a year ago, and we basically utilized methods that were established. After our pilot trial, we tweaked it a bit to optimize the quantity of metabolites that we were extracting to produce the best signal. So for the most part, they're similar, but we did tweak it a little bit. I didn't include any of the processes because it was just difficult to condense everything into 10 minutes.
Sure. Yeah, no, I understand. Thank you.
Yeah.
Thank you for that, Keaton.
Thank you.
We're going to go ahead and move on to our next presenter, Sitong Wu. And let me go ahead and get the presentation started.
Graduate student from Fresno State. Today I'm going to present my research topic, which is 3-carbomoyl 5,7,20 trimethylsilybenes synthesis and anti-proliferative evaluation. First of all, I want to introduce some background of my project. Prostate cancer has the highest incidence and the second highest mortality rate of all cancers in American men. Annually, 20% of all reported cancer cases are prostate cancer. Castration-resistant prostate cancer, which is CRPC, is the advanced and lethal version of prostate cancer. Of all diagnosed prostate cancer cases, 15% of them are identified to be CRPC. Current treatment include FDA-approved drug enzalutamide can only prolong patients' life rather than holding them due to drug resistance. So novel treatments for CRPC are urgently needed. Androgen receptor that is located in cytoplasm of the cell plays essential role in the development of prostate cancer. It initiates gene expression in cancer cells and promotes cancer cell growth by reactivating transcriptional activities in prostate cancer cells. So androgen receptor is a primary drug target in treating prostate cancer. The purpose of this project is to search for potential natural products derivatives that have anti-prostate cancer potency by inhibiting AR activities. A natural product, silybenin, that can be isolated from a plant milk thistle catches our eyes. It has a chemical structure like this. The earliest record on the medicinal use of migtistol for preventing and treating hepatotoxicity can be found in Hieronymus Buck's book that was published in 1539. In 1994, Dr. Agawa and his co-workers started the investigation on anti-cancer activities of silibinin, and in 1999, Dr. Lahiri reported that silibinin was able to prevent both stage 1 and stage 2 tumor promotion. Until 2001, Dr. Wen demonstrated that silibinin exhibits potency in preventing and treating prostate cancer by targeting androgen receptor. So silibinin is attractive to us because of its anti-AR activity. However, silibinin itself cannot be directly used as an anti-prostate cancer drug. It's a potential drug candidate because it has 3 potential problems. First, its anti-cancer potency is moderate. Second, it has low selectivity towards different prostate cancer cell lines. Third, its oral bioavailability is also low, which means silibinin in human's body is not stable. It can be easily excreted out of the body with metabolism, such as sweating. So these 3 problems pull out the objective of this project, which is to modify the chemical structure of silibinin to improve its anti-cancer activity, selectivity, and oral bioavailability. From 2019, we started this project to modify silibinin at 3' hydroxyl group by introducing carbomoyl group, which are some functional groups that have structures like this. We hypothesized that 3 carbomoyl silibinin derivatives will have improved anti-cancer activity, selectivity, and oral bioavailability. Generally, in order to test our hypothesis, we first synthesized desired compounds. Next, we need to use different spectrometry to confirm the chemical structure of synthesized compounds. Finally, we will test the anti-proliferative potency and selectivity of synthesized compounds towards 3 different prostate cancer cell lines, which are androgen receptor positive cell line, LNCAP, and androgen receptor negative cell line, DU155 and PC3. The IC50 values were collected from the WST1 bioassay. IC50 values means that the concentration of compounds needed to inhibit 50% of cancer cell growth. The lower IC50 values that the compound has, the higher anti-prostate cancer potency it has. Depends on whether or not the anti-proliferative potency of the compounds is high. We will decide if we're going to send them to our cooperators for further animal investigation. Specifically, 3 types of 3 carbomoyl derivatives of silibinin were synthesized by using either 3 or 2-step synthesis pathway. First, 3-step synthesis pathway was used to synthesize the synthesized compound 2, which is 3-carbomoyl 23-OTBS5720 trimethylsilibinin. The synthesis pathway starts with commercially available silibinin. Since our goal is to modify the hydroxyl group at 3' position, so the first thing we need to do is closing other untargeted hydroxyl group at 5, 7, 20, and 23' positions by introducing both methyl group and TBS group. And then I successfully introduced carbomoyl groups to 3' position by using DCM, which is a solvent, triazoamine, which is a base, and DMAP, which is a co-base. And when I tried to introduce carbomoyl group to 23 prime position by using the same method, I found out for the first time that the carbomoyl group can selectively introduce to 3 prime hydroxyl group when both the hydroxyl group at 3 and 23 positions are open. So we got synthesized compound 1. And I also collected a compound with both hydroxyl group carbomoyl as a minor product when I was running this reaction. At this point, as we can see in table 1, we synthesized 3 types of 3-carbomoyl cytidine derivatives. They can be 3-carbomoyl, 3-carbomoyl with 23 prime position protected with TBS group, and both 3 and 23 carbomoyl. 9 derivatives have been synthesized in 11 to 80% yields. Table 2 here showed the IC50 values of synthesized compounds collected from the bioassay. The red color number in the table indicated the synthesized derivatives have improved anti-prostate cancer potency compared to silibinin and enzalutamide. We can see that 4 out of 6 tested synthesized compounds which have been marked with stars can selectively inhibit androgen receptor positive LNCAP prostate cancer cell proliferation with IC50 value less than 1 micromolar. In conclusion, we found out for the first time that we are able to selectively introduce carbomoyl groups to silibinin at 3 prime position with the mediation of triethylamine and DMAP. The bioassay data revealed that 4 out of 6 tested derivatives can selectively inhibit AR positive LNCAP prostate cancer cell proliferation with IC50 values less than 1 micromolar. And finally, 3-carbomoyl silibinin derivatives may target androgen receptor, but we need to verify this in future investigation. Here are my references. I'd like to say thank you for the support of chemistry department at Fresno State and the College of Science and Mathematics. I also appreciate CCRS to give me this opportunity to be here. And thank you for your listening.
Thank you for that, Satong. We're going to go ahead and get started with the Q&A.
Any questions?
I do have one that came through. I'm monitoring the live chat, and somebody did ask a question on the live chat. So I'm gonna go ahead and read it. It says, are mRNA vaccines like the ones that were developed for COVID-19 prevention, implementation, or planned for implementation in the current cancer research?
Yeah. So can you repeat the question again?
Yeah.
Because
yeah, for sure.
Yeah.
So they're asking, are mRNA vaccines like the ones that were developed for COVID-19 prevention implemented or planned for implementation in the current cancer research?
Okay. Yes.
Yes. Okay.
So yeah, Dr. Brooks.
Great, thank you for the interesting talk. I'm just kind of curious a little bit more about the bioavailability question.
Oh yeah. Okay.
What kind of chemistry do you have to do to make a compound more bioavailable?
So in order to make it more bioavailable, like silibinin, it's very unstable in human body because there are like too many hydroxyl groups in its structure. It's not stable. And so we are trying to introduce some chemical ligands into the hydroxyl group and close the hydroxyl groups to make it like insoluble. Then it's gonna be like stable in more stable in human's body and improve its oral bioavailability, and it won't like easily excrete it out of the body.
Okay, thank you.
Yeah, Dr. Uchida.
Hi. So I have a pretty naive question. You said, one of the problem of silibinin is low selectivity toward different prostate cancer.
Yes.
Then I can see if there's low selectivity between just a normal cell versus cancer cell, that could be problem. But why low selectivity toward a different type of prostate cancer is also problem? I simply thought if they can treat any type of prostate cancer, that would be great. But why that's problem?
Yeah, but we wanted to be like more like focused on one type of prostate cancer. Like I mentioned, the castration-resistant prostate cancer. That is our goal. Yeah.
So then if the compound has the less selectivity toward two different type of prostate cancer, that can be the problem?
I won't say it's a problem, but like I said, our target is like AR positive, like the cells with androgen receptor. So we want our compound to target like AR positive prostate cancer cell lines, because androgen receptor is our target.
Okay, thank you.
You're welcome.
Thank you for that, Sitong. And we're going to go ahead and get started with our last presenter for this morning session. It's Angam Ahmed. Am I saying that correctly?
Yes, you are.
Yes, thank you. Okay, I'm going to go ahead and get it started.
I'm Ahmed, and today I'm going to talk about my project, which is called Structure of Enterohemorrhagic E. coli O157H7 Intimin virulence factor bound to nanobodies. E. coli in humans and mammals GI tract aids in the digestion and absorption processes, as well as vitamin K production and maintaining the balance between good and harmful bacteria. However, there are hundreds of strains of E. coli that exhibit different characteristics. For example, enterohemorrhagic E. coli is a food and waterborne pathogen that normally resides in the intestinal tract of cattle as a normal flora. However, it can be transmitted to humans through the ingestion of contaminated food products, or from human-human contact, or a human-to-animal contact. There are 7 serotypes of EHEC. 6 of them are called Big 6, and 1 of them called O157H7. This one is the most virulent of all of EHEC strains and is associated with epidemic outbreak. Going on to the mechanism of infection of EHEC is that it involves 3 virulence factors: TER, intimin, which is the main focus of our research, and Shiga-like toxin. As you could see here, once EHEC invades the host GI tract, it starts secreting the receptor TER into the host cell membrane. Then it uses intimin to adhere tightly to the host cell membrane, as you could see here. In the zoom in picture, this is EHEC cell membrane, and this is the host cell membrane. And this shows intimin to interact with its receptor TER. So for our study, we're not interested in studying the full protein, but actually 3 domains, sorry. And they are made of 277 residues. So once EHEC is tightly attached to the host cell membrane, it starts secreting a potent toxin called Shiga-like toxin, which can lead to a serious and potentially fatal GI infections. Currently, there is no treatment or vaccine against this bacteria besides the use of good hygiene. However, patients with critical conditions are put in intensive care units where they receive blood transfusion and kidney dialysis. One possible solution to prevent EHEC pathogenesis is through the use of nanobodies or single-domain antibodies, also known as VHHs. These antibodies are naturally produced by Camelida family, which includes llamas, alpacas, and camels. They represent 30% of entire antibodies in their sera, in addition to conventional antibodies. As you can see here, this is a conventional antibody, and this is a heavy chain only antibody. When I talk about nanobodies or VHHs, I'm referring to the antigen binding domain of these antibodies, and that's what we are working with, just that domain. So these antibodies exhibit superiority over conventional antibodies in terms of their small size, which allow tissue penetration, as well as high pH and temperature range. So we have 6 antibodies that nanobodies that we're working with, and we obtained them from our collaborator at the National Research Council of Canada, from Dr. Henry. What he did is that he injected a llama with Intamin 277 fused to maltose binding protein, which is a fusion protein. And they collected the blood, extracted messenger RNA, and then amplified it for PCR, and cloned it to a fragment expressed these antibodies or nanobodies, and selected for the high specific nanobodies against Intamin 277. And then they tested the affinity by ELISA. As you can see here, we have 5 nanobodies against Intamin 277 and 1 against maltose binding protein. The hypothesis of our research project is that Intamin-specific nanobodies show high specificity and binding affinity for Intamin. Therefore, we hypothesize that these nanobodies would have the capacity to neutralize EHEC pathogenesis. The goal of our research project is that to determine the complex crystal structure of these nanobodies bound to Intamin to unravel their binding mechanism using a method called X-ray crystallography. So this is the scheme of our experimental process, is that after we obtain the plasmid containing the genes of our nanobodies as well as the antigen, we transform them to E. coli, and we purified the protein from E. coli and characterize it. So once we have a high quality protein, then we complex the antigen to the nanobodies. We co-purify the complex using size exclusion chromatography, and then we set up crystal trays using a 96 different buffer conditions. Once we have crystals formed in one of these conditions, we grow large crystals using the method called hanging drop diffusion. And once we have large crystals formed, then we send them out to CLS, or Canadian Light Source, for X-ray diffraction data collection. Then Dr. Brooks processed the data, and then we solved the structure using a software called Phaser. Then we analyzed the structure in another software called PyMol. So we determined the structure of one of the nanobodies called VHH1 bound to Intamin 277. As you can see here, yellow and green are for the antigen Intamin 277, cyan and salmon are for the nanobodies. So the nanobodies form a polar contact with the antigen using CDR3 as well as framework 2, as you can see here. Also determine the structure of VHH2 bound to Intamin 277. As you can see here, again, yellow represent the antigen, cyan represent the nanobody, and it shows that this nanobody interact with the antigen via polar contact using framework 2 as well as CDR3, and also CDR2 that shows to bind to another copy of Intamin 277. We didn't know if that's true or it's a crystallographic artifact, so we need to do further experiment to confirm that. In addition, we determined the structure of VHH3 bound to Intamin 277 Also, again, yellow represent the antigen, and cyan and salmon represent the nanobody. This nanobody again interact with the antigen through polar contact using framework 2 and CDR3. So we perform also epitope mapping to see if these nanobodies are identical in their binding in terms of their residues, and we see that VHH3 and VHH1 are almost identical except for one residue, alanine 28, but intimin or VHH2 is slightly different with two residues. Our data confirms initial results obtained by our collaborator, Dr. Henry, where he performed SPR epitope pinning to show if these nanobodies bind on the same epitope or at different epitopes. He showed that the three of the VHHs bind on the same epitope. One of them binds a little different, and VHH5 binds a totally different epitope. Our findings were unexpected, meaning that when we superimposed the nanobodies bound to intimin 277 to a previously determined intimin structure bound to TER, we show here that these nanobodies interact through CDR3 and also framework 2. However, they do not seem to bind to the location where intimin binds into TER. So we think that these nanobodies do not inhibit EHEC pathogenesis. However, VHHs exhibit unusual mechanism of binding. As you can see here, these nanobodies use their framework to bind into their antigen. So future direction is that we're going to transform and express TER protein and perform a competitive ELISA to see if these VHHs overcompete TER for intimin. We're going to mutate framework 2 residue of VHHs, as also do diffraction data collection of VHH4, VHH5, and triple complex and determine their crystal structure. So I would like to thank Dr. Brooks and Brooks Lab for the guidance and support, as well as Dr. Henry at the National Research Council of Canada, and the FSSRA and Sally Casanova for their support for my research project. Thank you.
Thank you for that, Anna. I'm going to go ahead and open it up for questions.
Okay.
Hi there. So I had a few questions about the crystal size requirements for crystallography. You were saying that you grew large crystals. Is there a lower limit for the size that you're aiming for to collect useful crystallographic data?
I think it has to be larger, or around 100 micrometer, I think, if I remember correctly.
Okay.
But to be diffractable.
Got it. And then as a follow-up question to that, were the structures that you presented obtained based on data from single crystals, like one crystallographic measurement on a single crystal?
You mean for each structure?
Right.
Yes. For each structure, it's from one crystal.
Okay. And would there be an advantage of collecting replicate measurements? Because you were saying in your talk that there may be like crystallographic artifacts that came out of the data. Would there be an advantage to collecting data using multiple different crystals, like replicate measurements?
You mean using different crystals of the same protein?
Right.
Yeah. As long as you get like a good diffraction data and good resolution.
Got it. Okay, thank you.
You're welcome.
We have a question from Dr. Uchida.
Hello.
Hi.
So I think this is not probably directly you are part of the this research, but more likely the collaborator's part. But I'm curious, the collaborator probably used this phage display library in your, according to your slide.
Yeah.
Then I assume the length of this phage library is probably like a 10 or 12 amino acid in that it's really flappy part. Then in your nanobody, this amino acid is, has the three-dimensional structure. So is it just fair to compare these really, random or certain amino acid sequence at the end of this phage? It could be really flappy versus your nanobody that is more structure, is rigid. Do you have any comment or some thought about that?
Are you talking about like the sequence of the?
Not just the sequence, but in terms of the structure, dynamics versus statistics.
I honestly don't know.
Okay. Thank you.
Yeah.
Thank you so much for all of your participation. And to all the judges, thank you also for all the participation. I'm going to go ahead and start closing down the session. We welcome you to join us for any of the sessions later on today. And I hope you guys have a great day.
Thank you.
Thank you.
Thank you.